So good afternoon, everyone. Today we will be discussing about medical virology. So when we say medical virology, we will only be uh, concentrating two viruses that are of medical importance. Other than medical viruses, we will not be tackling any of those. So for starters, what is a virus? Viruses or virus are considered as the ultimate parasites functioning only within another cell, meaning to say exposure of virus outside the cell for a longer period of time will lead to its inhibition or killing of the said virus. Because virus can only grow well, protect themselves once inside the cell. Otherwise, they are very much um, exposed to other factors that may lead to their untimely death. So they do not grow larger and divide like other organisms. They attach to and enter the host cells, then direct the cells to make viral components. When we say viral components, these are the new viruses that are assembled from these components. Viral size are normally ranges from 10 nanometers to 300 nanometers in diameter and can only be observed in an electron microscope. If you can remember your electron microscope, you have the scanning or the transmission electron microscope, the SEM and the TEM. However, some research laboratories that studies about virus can also have microscopes that are the same of the compound microscope. However, it is not the morphology of the virus they are studying, but the cytotoxic or the cytopathic effect of the cells which are being observed in the cell lines. Cell lines is spelled as CELLLINES, cell lines. Okay. Please take note that no type of organism is safe from viral infection. Why is that? Even bacteria, protozoa, other parasites, fungi, including other viruses, can be infected by a virus. Okay, you have the 5 specific properties of the virus. First, they possess either DNA or RNA, but never both. Okay, so your virus, it's either a DNA type of virus or RNA type of virus, but not both. They are unable to replicate or multiply on their own. So replication is directed by the viral nucleic acid once they are introduced to the host cell. Unlike most cells, they do not divide by binary fission, mitosis, or meiosis, and they do not have the enzymes necessary for energy production. Viruses depend on ribosomes, enzymes, and metabolites of the host cell for protein and nucleic acid production. That's why viruses, as of this time, are still on debate if the virus is considered to be a living organism or a non-living organism. However, most proof or documents tend to point out that viruses are more of a non-living organism rather than the living kind. Another unique characteristics of viruses are that they are energyless. The basis life form composed of a protein material called capsid that surrounds a genetic material. Their genetic material is either RNA or DNA, and the replication of genetic material occurs when the virus takes control of the host cell's synthetic machinery. Okay, so what are the components and structures of the virus? First, ang pinaka-basic it's they are just two. First, the genome, which is either an RNA or DNA, and of course, the protein that encloses the genome, which is termed as nucleocapsid. Your nucleocapsid could also be icosahedral or helical in morphology, or somewhat complex. Okay? Some virions possess a cell membrane-derived lipid envelope containing viral glycoproteins that surrounds the nucleocapsid. Your viruses with envelopes are referred to as envelope viruses. Otherwise, they are called as naked viruses. The purpose of the envelope in envelope viruses is to protect the genome and the capsid. But at some point, naked viruses tend to be more resistant to disinfectants rather than with envelope viruses. Okay? So capsids and envelopes serve to protect the viral genome in its extracellular state from physical and enzymatic destructions. Capsids containing binding sites for attachment to cell receptors and mediate in entry to the virion of the virion into susceptible cells. Take note, guys, had the term cell receptors. Cell receptors. Your virus cannot infect a certain cell if it does not have the cell receptors. It only means that your viruses are site-specific or cell-specific. Okay? Your hepatitis could be found on your body fluids, but it's only the hepatocytes that it can infect. Your hepatitis virus can travel to the brain, but it's just the hepatocytes which they can damage. Okay? Some viruses contain enzymes like your virion polymerase or other viral proteins necessary for efficient viral reproduction. The viral nucleic acid genome encodes information needed for viral replication and the synthesis of progeny virions. The nucleic acid of the virion or the genetic material could be single-stranded or double-stranded, linear or circular. Your DNA viruses genomes may be single-stranded or double-stranded, linear or circular molecules. Your RNA viruses genomes may be single-stranded as well, double-stranded, linear or circular, but may subkon pa, either segmented or non-segmented. Okay? So the viral structure or the nucleocapsid has two types of symmetry, either helical or icosahedral. The capsid symmetry is based on the arrangement of the morphologic subunits called capsomeres. Capsomeres are composed of multiple copies of one or several polypeptides that forms your capsid. So your helical nucleocapsid are composed of protein subunits bound to the viral RNA in the form of a helix. Helix are normally found on RNA viruses. Your icosahedral nucleocapsids have protein subunits arranged in the form of icosahedrons with 20 triangular faces and 12 vertices. The viral nucleic acid is located inside the capsid shell. So construction of viruses with icosahedral and helical symmetries is the most efficient and economical means for assembling a virion with limited coding capacity. Some viruses, however, have neither helical nor icosahedral symmetry and are designed structurally as complex virus. That's why, as I mentioned earlier, your nucleic acid could be RNA or DNA, the genomic material. Your capsid could either be symmetry, helical, icosahedral, or complex. Okay? So the viral morphology of the nucleic acid of the genome, again, it is either classified as RNA or DNA, and it can be a single-stranded, double-stranded, linear, or looped in separate segments or in one continuous strand. The nucleic acid sequences can encode a simple message or encode hundreds of enzymes and structural proteins. So the RNA viruses has three types, either positive stranded, negative